I'll just use this. So I always mess something up. Yeah, these are first. All right, great. So thank you very much for inviting me to come to this meeting. It's been really stimulating and a wonderful blend of people doing many different things. I've learned a lot. So the title of my talk today is Becoming What You Smell, Adaptive Sensing the Olfactory System. You'll see what I mean by that as the talk progresses. So I'm a relative newcomer thinking about the sense of smell. So what I find intimidating about thinking about olfaction is how difficult it is. So the space of smells is really intimidatingly diverse. If you ask people how many volatile molecules are there, conservatively people will tell you 10,000, some people will tell you 100,000, occasionally good with a million. That's a vast number of molecules that you may wish to sense. To make matters worse, typical natural odors, let's say from a flower and so on, are mixtures of at least 50 molecular species, especially in sort of foods and flavors and that kind of thing. So, you know, conservatively, there are 10,000 to the power 50 natural smells. That's a huge space of sort of objects, of odor objects that you might wish to sense and represent. To make matters even worse, the odor environment changes, reflects changing seasons, circumstances, new opportunities, new threats. So it's a really very difficult problem to be able to sense and represent this kind of information. So olfaction, thought about in this way, is kind of a system for feeling the shapes of molecules. Right? You've got many molecules, you've got a mixture of them, you've got to feel them all and figure out what it is that's coming to your nose. So this problem becomes especially hard because while in this language the odor space is high dimensional, using a different language from the language that Tanya was using yesterday, right? Let's say 10,000 dimensional, the representation space that you have in your nose is very low dimensional. It's perspective. This is because you sense molecules when they bind to receptors. Every receptor is encoded by a separate gene. And if you think about functional odor receptors, in fly there's sort of an order 100, in human there's order 300, in mouse there's order 1,000, and elephant there's order 2,000. Right? They're sort of order of magnitude. They're all like from fly to elephant, you have a few hundred receptors, give or take a few, a factor of a few. It's a very low dimensional response space in which you're supposed to represent this information. You could say, how could you possibly represent such a high-dimensional odor space with so few responses? Well, so you might make the analogy, suppose I forced you to represent three-dimensional space with one coordinate. Could you do it? Well, of course you could. You kind of run a space-filling curve through the 3 dimensions, read out the coordinate, but that's a really stupid representation because nearby points in 3 dimensions will get mapped to distant points along this line, and why do you put things into your head anyway? It's so that you can tell what's similar and what's different. It's going to get very hard if you use a representation like that. In other words, there's a required invariance in the representation. You would like, if possible, to find this low-dimensional version of this high-dimensional odor space in such a manner that you preserve, in some sense, the distances between things so that you can make judgments of similarity and difference. So can you do this? Well, so actually, it's not just olfaction that faces this kind of constraint. It's the problem of life. You have to do a lot with a very little, and we see this kind of problem again and again. So in vision, there's a version of this sort of limited resource problem, and the standard answer that's given by vision researchers to how you deal with the problem of having too little to see the world is that you adapt to the environment in different ways. So for example, real visual scenes, natural visual scenes, have structure at many different scales. At the high level, the world is made of objects of different kinds, edges and corners, and baboons and trees and what have you. Those are composed indeed of edges, corners, etc. And we've heard a lot about that in the last couple of days. At the low level, there are also regularities. So for example, if you look at any color band, red, blue, green, for example, you'll find that the distribution of light is log normal. Right? So that's a regularity in the world. You'll also find that the long-range correlations, here is a log-log plot of the power spectrum, and falls in a straight line, implying that pairwise correlations of light in the natural world are scale invariant. So famously, there is a theory of early vision, at least, that says that what you should do to deal with a world like this is you should tune the fixed structure of circuits to these regularities, and then of course these things change a little bit in light level changes from dawn to dusk, what have you, and so then you adapt the circuits in an ongoing way in order to commit your limited resources as effectively as you can manage. So in vision, this is supposed to give us a theory, an explanation of many, many, many diverse phenomena and aspects of the structure of circuits. So, for example, the fact that receptor fields in retina are center minus surround, receptor field spacing and organization, the edge detectors in V1, color opponency, the excess of off cells, namely dark spot detectors over on cells, namely bright spot detectors, the visual salience of textures. These are very diverse things. And many of these, and they range in their processing level from the retinal periphery all the way to some layers of the visual cortex. Many of these things have been at least claimed to be explained by adaptation to these kinds of high-level and low-level structures in the visual world. So we may ask the question whether we can repeat this trick to try to understand some of the interesting features and the very hard information processing problem of olfaction by thinking that, well, the only thing you could do is adapt the circuitry to the structure of the world, and we can ask what consequences that has. Today, I'm going to try to tell you about 4 stories in that regard. The first one is going to be about the sensory periphery and how you take such a high-dimensional space and store it in a low-dimensional space while preserving distances. Then I'm going to tell you about, once you've done that, there's some ongoing adaptation you can do to improve the representation. So I'll talk a little bit about that. Then I'll try to get serious with the actual sensing mechanisms, the fact that real receptors are not linear sensors of things, they're nonlinear. And then I'll try to talk a little bit about how you might decode such a fancy combinatorial representation of odors, which is what we're going to find. Okay, so I'm going to try to get through that, and if I don't, I don't. Okay, so let's start. So I'm going to start, this is work done with Anne Hermanstad, Tamesh Krishnamurthy, Alexandra Balchuk, and Thierry Morin. Okay, so once again, here's our problem. We have a high-dimensional odor space. We have a low-dimensional representation space. We wish to represent points in these high dimensions, in these low dimensions, while preserving distances between pairs if we can. How could we do this? Well, we need to adapt to some sort of structure in the space of odors. So what structure is there that you could use? So one idea is that there's an interesting kind of structure that odors are actually sparse in a certain sense. If you have here all the volatile molecules and here all the odors, odor 1 may contain this molecule and this molecule and this molecule, but not all the others. It contains only a few molecules out of all the ones that could be there. That's because your typical animal or plant can't really afford machinery to make 10,000 different odorants. It's going to make like 50, 100 maybe, right? The things that are perceptually important seem to be about of order 50. So, in this sense, the geometry of olfactory mixture space looks like this. If I'm drawing here, these 3 dimensions are supposed to be the space of all possible odor concentration vectors. I pick a concentration for every molecule. The actual odor space that you deal with, the olfactory objects, are this green, red, and blue, and they fill out, in this case, let's say this line, this line, this line. So although you could be anywhere in these 3 dimensions, your olfactory objects, we can promise, will live on this line, and this line. In other words, the space of objects that you wish to represent is a kind of union of k-dimensional hyperplanes, planes, in some n-dimensional space of volatile molecules, if you take this perspective. Okay. So odors, of course, complex, which means that the number of components, k, in each odor is much bigger than 1. On the other hand, they're sparse or simple because the number of odorants, k, is much less than the number of odorants you could have been using. Okay, so we want to, let's ask if we can exploit this to produce a nice low-dimensional representation of this information. So if you were to ask a mathematician that, or a signal processing person, they'll say, why, yes, you can do this. Because maybe some 10, 12 years ago, there was the idea of something called compressive sensing, which goes like this. Suppose I tell you the data you wish to represent are very high dimensional vectors, n-dimensional, but sparse. That is to say, I promise you that there are no more than k non-zero entries, which indeed sounds a little bit like what odor object concentration vectors look like. So there's a rule, there's a theorem that says, now you can multiply this. This is linear sensing, not non-linear sensing. You multiply this by a matrix, a sensing matrix, which is rectangular, so that the number of rows is much smaller than the number of columns, n by n. And now from the sparse high-dimensional vector, you get a dense low-dimensional vector. And if you pick m to be of order, the sparsity k, have the logarithm of the dimension of this big vector, there's a theorem that says, well, actually, you store all the information faithfully in a small vector. Right? So there's a decoding algorithm that allows it to recover the high-dimensional vector from the low-dimensional vector. So it's faithful. What's more, it's a bit like magic. The distances in odor space get preserved. So if you have two high-dimensional sparse vectors with some distance between them, and you do this trick, right, you will get a low-dimensional dense vector in which the distances are the same. Right? So that's kind of like great. That's exactly what the doctor ordered. And looking at this, you would say if evolution hadn't used this, it would be stupid. Right? So we should go looking for this as an explanation of some kind of structure that may be there in the olfactory coding. Of course, you should ask me, what should this sensing matrix look like? Does it need to have any special properties? And the answer is yes. There's a property called the restricted isometry property, which is too complicated to explain kind of thing. But an example of that of a matrix with that property is a random matrix here. If these are really binding affinities of receptors to odors. If you pick them randomly, it'll do the job. Right? For me, that inverts the way I usually think about receptors. Really, I often think about the problem for receptors is how they evolve and co-evolve and change with the molecule they're trying to sense, so the binding is strong and good, efficient, doesn't bind anything else. But here, to get this kind of thing to work, what you really want is sort of floppy receptors that bind to lots of stuff in a kind of randomish kind of way. That's what you'd wish. So, well, we may hypothesize then that an interpretation of how olfaction works is that each receptor binds with, quote, random, sort of, affinities to volatile molecules, and then, given the sparsity of natural odors, you should expect that, you know what, a few hundred receptors should do you fine to represent any odor. So I'm going to try to investigate this. So to do that, the strict mathematical theorems, sadly, work with linearized sensing. So the first thing we're going to do is to linearize the actual sensing mechanisms in the nose. The way I'm going to do that is here is, for example, mouse receptor OLFR895. You can measure dose response curves. And these are not linear. They go up like this, they're non-linear. But we're going to linearize them around an operating point. We'll just sort of draw straight lines here, here, here. And that gives an approximation of how the receptor works in this region. And so that's what we're going to do. Right? So we'll start out like that. I'll correct that in the third part of the talk and tell you how to deal better with the actual nonlinearities. The real problem being how do you even characterize them in the first place. Okay, so we're going to take this sort of linearized version of the sensing. And here's my schematic of the sensing model that we're going to do for Drosophila. We're going to do Drosophila, I should have said. It's going to be like this. I imagine that there are, really this is written out from mouse, but anyway, there's a bunch of receptors, different kinds. There are olfactory sensory neurons that have different kinds of receptors in color. The way the olfactory pathway works is that receptors of each kind, like say the red kind, are collected together in a second structure called a glomerulus in the olfactory bulb, and they're sort of summed over, presumably to sort of partly reduce noise and so on. And then there's a bunch of subsequent processing to the cortex, which we're not really going to talk about today. So schematically, here's an odor vector, concentrations of different molecules. They get sort of multiplied or sensed by the sensing matrix. Really, we mean that there's a linear response function controlled by various affinities, and that leads to various patterns of activity in these glomeruli, which are supposed to be the output neurons of the sensory periphery. Okay? So we're going to do this with measurements done by Hallam and Carlson in Drosophila. Here is a sensing matrix. Here are, actually, olfactory receptor neurons should be here. There are 25 olfactory receptor neurons and 100 odors that they investigated. And so they got this matrix of affinities between different receptors and different odors. Now, of course, this is hardly a complete map of the olfactory world. There are maybe 10,000 odorants, but it just takes too long to study all of them. Likewise, even Drosophila has about 60 receptor neurons, so this is a subsampling of them. But we'll take it because that's what we have, and we'll try to get off the ground with that. So what I'm going to do is I'm going to pretend that this is a compressive sensor. And I'm going to try to decode the code as a means of assessing its quality. So here we go. So this is this measured sensing matrix. We agree that I'm going to randomly generate odor mixtures of different complexities with different numbers of odorants. I'm going to sense them linearly with this sensing matrix, and then I'm going to use the mathematician-approved decoding algorithm to decode this to see, can I get back information? That isn't to say the brain uses this decoding algorithm. This is an assessment of whether or not this code would preserve the information. We'll get back to an actual decoding algorithm later. The green line here shows you the performance that you get using the actual sensing matrix. You'll notice the actual sensing matrix doesn't look random. There's certainly correlations in this. So the first thing you could ask is, suppose I made this a little bit more random, does it do better? So you could sort of scramble it. And if you do, you find that it doesn't do any better. Right? So the scrambled version of the actual sensing matrix gives you similar decoding performance. Then you could ask, if you instead interpreted the sensing matrix as really not being all of these numbers here, but that you threshold it, and that should only pay attention to the strong responses, the weak responses are really just noise, right? You should ignore them. Then you would get something that looks more like this. This is basically a thresholded version of this. Right? So you keep only the strong responses. And then you find the decoding performance is much worse. And finally, you can compare with an ideal Gaussian random sensor, in this case. And you find that the performance is pretty comparable. Okay? So that seems hopeful, but perhaps I should interpret this in this way. So the next step is to really ask, suppose I try to extrapolate this, and I have this sensing matrix, and I want to extrapolate the sensing matrix with the same statistics to make a much bigger one that it responds to 10,000 odorants, let's say, right? I'm going to try to build a bigger sensing matrix responding to 10,000 odorants and with 60 receptors, 100 receptors, etc. to model the way real. I want to keep these kind of statistics because I assume these statistics reflect the statistics of sensing, right? But I want to make the thing bigger to reflect the complexity of what actually happens. So we have a method of doing that, and I'm not going to belabor that. These are just various checks, eigenvalues of covariance matrix, response histogram firing rates. So I'm going to guarantee to you we can develop a larger artificial sensing matrix that preserves what we believe to be the essential correlation statistics and the response statistics of the actual measured data. So I'm not going to go into the detail of exactly how we did that. So with that, we can now go ahead and measure the thing we really wanted. What we want is we want to have a measure of distortion where we take high-dimensional but sparse odor vectors, sense them with these sensing matrices, right? And then ask, do you preserve distances? Because that's what we said is the invariance you should probably preserve if you want to later decode faithfully similarity and differences. So here's a picture where you take 50 component odors, the supposed complexity of natural odors, and you construct various of these sensing matrices. The colors indicate, well, suppose, I'm not really sure when someone gives me one of these sensing matrices where I should put the threshold for saying it's responder or not. So I'm going to put the threshold at various levels. That'd be 20% of the things that are considered responding, 40%, 60%, etc. And then what you can do is you can measure the distortion as a function of the number of receptors involved in your model olfactory system. And what you find is by the time you get to a couple hundred receptors, you get lovely performance, and there's very low distortion of most of the odors that you present to it. The distortion is low, and what's more, you can make the responses pretty sparse, so that regardless of what, there's a robustness to where you put the threshold for interpreting whether the neuron has fired or not. And we can do this another way. We can instead pick, let's say, a couple hundred receptors, and then repeat this analysis. And then what you find is that with a couple hundred receptors, no matter how complex you make the odors, up to about 200, 300 components in the odors. These are very complex odors, like coffee is a few hundred components, right? So these are really complicated odors. The distortion is small, so you should hope that any reasonable decoding mechanism will be able to separate similarities and differences pretty well. To me, this is, first of all, really cool and amazing. But secondly, this makes me wonder that perhaps this kind of finding about distortions explains the operating point at which the olfactory systems of all animals seem to sit. From fly to elephant, it's a few hundred receptor types, give or take a small factor. And maybe that's because with a few hundred receptors and the complexity of actual natural odors, you can actually produce a low-dimensional representation with low distortion of distances. Okay, so now I'd like to think a little bit more about that, and I'm going to think about that in the following way. So far, what I've been trying to say is tell a story about the adaptation of the gross structure of the sensing in the olfactory system to the gross structure of the olfactory world, the sparseness, right? But now I want to think about, well, in detail, you have these receptors, and they bind in this sort of broad way with a certain sparseness and so on. Now what I want to think about is, well, when you actually experience the world, in fact, in the fall and the spring, the smells are in fact different, right? And smells change at different times. So given that you have a certain receptor repertoire, what could you do to improve or better sense the world, given that whatever plasticity you have left, given that you've already genetically encoded all the receptor types? What's that? Oh, sorry. Yeah, sorry. You weren't telling me 5 minutes left. Right?
No.
Oof.
I was just going to ask a question.
Yes. Oh, good. Excellent.
Which is in the first part, as you said, if you use compressive sensing, you need a certain coherence or lack of coherence, and your matrices therefore have to be appropriately sufficiently
Absolutely.
random. And you also measured them. Is there some way to test?
Yeah. So, they obviously have correlations, and they're not random in quite the way that you expect. You need the incoherence you need the restricted isometry property, which you need all the spectra. So these are not entirely incoherent like that. And indeed, their performance is a little bit worse than it would be for something that actually satisfies the property. Let's take the Gaussian random matrix, for example. So it's not as good. But then I would say that actually, there are two parts to the answer to that. One is I'm not sure actually how to quantify how far you are from that property, other than by testing the outcome, which is how distorted the vectors are. So that's why we have a distortion measure here. Another thing I would say that if you think about the evolution of receptors, again, I had always thought about how do you evolve receptors that are strongly binding to this pheromone or this I don't know, something like that. But here the issue is, if you believe this picture, that the selective pressure should be for randomness in the receptor. It's actually not that easy, if you think about it, to build a random receptor. Because if you bind to this small, if you bind to a large shape, then you'll be very specific. If you bind to a small shape, then well, you'll bind to whatever has that small shape and not to anything that doesn't. So there'll be correlations because of the nature of molecules. So it's actually hard to evolve, I think, a random receptor. But I don't fully understand this yet. I'll take one more and then I would like to go on.
So your strategy sounds very much like immunology, where the first effects are non-specific or quasi-specific, and then you go to more specific.
So I couldn't agree more. Partly because I started working on olfaction. I've now written several papers on the adaptive immune system. But I would like both in vertebrates and in mammals.
I just want
right. So, but there is an analogy, and it's an interesting one that I don't have time to go into it. Happy to talk about it afterwards. Yeah. So that's true. So next I want to talk about receptor abundances. Why is that? I said, well, what can you adapt once you've committed to a certain repertoire of receptors? What can you adapt? Well, the thing that's left is the abundances of different receptors. Now, receptors are expressed in the nose in olfactory receptor neurons, and usually we're told that in adults, there's no neurogenesis by and large. But one of the places where there is neurogenesis is the olfactory periphery. Every month, all of your olfactory receptor neurons die off and are replaced. And they're replaced, and it turns out that there are a number of papers by various people, including Catherine Dulac's lab here at Harvard, which have shown that the proportions of the different receptors that are expressed vary with olfactory experience. So for us, this is an opportunity, because then you might say, if I'm taking this sort of adaptive view, that it's basically you're shaping the olfactory system to the world, maybe this is an adaptive remodeling. You might phrase the question a little bit more precisely like this. Given a natural odor distribution and a set of receptor types and particular tunings, how should you pick the fractions of different receptor types that you express in order to sense the odor environment well? Now, okay, so we have many problems with trying to do this. One is we don't know the natural odor environment. We don't really know the binding affinities of all the receptors and everything. So how do we get off the ground? So a way of getting off the ground is to hope or suppose that you're trying to extract something more universal as opposed to detailed in specificity, some general pattern. And then you can imagine that you're going to model the olfactory environment in terms of the mean intensities of the different odors and their covariances, right? There's this odor object and that odor object, these things vary, the molecules here and there vary together, and there's some typical intensity, that kind of thing. And again, I'm going to take, for the moment, a very simple linear sensing. You take the concentrations, you multiply them by the sensing affinities, you get the response. But now I'm going to get a bit more serious. I'm going to agree that there's some noise and has some variance, right? I've been really focused on that earlier. But then I'm also going to agree that you give me, for each receptor of type J, you give me kj receptors of that type. There's a certain number of receptors of each type. And so when that signal from these receptors is pooled to the olfactory bulb, we might suppose that it beats down the noise. Right? This is a simple and somewhat crude model of exactly what goes on. But we're going to try to get sort of qualitative results out of it. And you'll see they're actually semi-quantitative. Okay. So once again, here's our sensing model. Receptors pooling into the so-called glomeruli. Environment vector is sensed by some linear sensing matrix plus Gaussian noise. And because I have chosen Gaussian everything, there's an analytical answer to the mutual information conveyed by the output of these receptors about the input. So this is the analytical answer, and that's what I'm going to work with. But of course, in the actual papers, we also did a lot of numerical studies that didn't involve Gaussian assumptions about everything, and you can recapitulate and see that the things I'm going to say basically hold out, with some quantitative differences if you make things non-linear and so on and so forth. Okay, so this is the answer for the overall mutual information. So for those of you who know information theory, this should be reminiscent of the classic Shannon Gaussian channel trace 1 plus log of signal to ratio. Log of 1 plus signal to ratio. Here's why. So if you look at this, the K here, this is a diagonal matrix of the receptor abundances. So the higher the receptor abundances, the more information you should be getting because you should be beating down the noise. Sigma here is a diagonal matrix of the noise variances. So the more the noise, the less information you should be getting. And then Q here is a matrix that's really the covariance of the responses. So if this was diagonal, this would be precisely the kind of thing we're familiar with in conventional information theory. You have a bunch of independent responders, sensors. Each one has some signal variance and some noise variance. And basically the signal to noise ratio, the ratio of those variances, should tell you how much information is conveyed. The key point here is, in fact, these are not diagonal. That's because the receptors are all going to be correlated with each other. Here I'm trying to deal with the fact that there are actually correlations. Okay. So we need to ask, what is the consequence of having these correlations? And I'm trying to summarize that in one figure here. That it turns out that if you look at the actual abundance of receptors, what they turn out to be correlated with is the diagonal elements of this inverse of this overlap matrix of the noise variance. I'm not going to go through that in detail, but we can expand that. But the key issue here is that if I take Q, this matrix, and I invert it, then the diagonal entries, which wind up determining the abundances of things, are sort of depend globally on all of the correlations between every receptor and every other receptor. Let me say qualitatively why this should make some sense, right? The reason is, suppose you have this receptor that responds a lot, but its response is correlated with many other receptors. So it's unclear whether you're going to get the more information from the response of 999 weakly responding receptors or one strongly responding receptor. It may be better for you to actually use those 999 weakly responding receptors. You may get more out of that to the collective effect. So that's what's really going on here, but there's a sort of highly non-local dependence on what's best on the overall correlation structure of the responses. Okay, this has various consequences. One consequence is if you're thinking about a olfactory system with lots of neurons, lots and lots of neurons, then this would predict that you have high diversity of receptors and that the receptors are sort of more evenly expressed. Lots of neurons is equivalently high signal to noise in this case. If you have very few neurons or low signal to noise, you get the reverse as a prediction, that what you should find is low diversity of receptors and highly inhomogeneous expression of receptors. So in fact, that's at least a qualitative statement here, because we're dealing with a qualitative model. You can sort of compare that across species. This is actually hard to do, because you'd be surprised how little data there is on the actual receptor repertoires across species. So here we're doing, allometric scaling, and there's some results about how the size of the olfactory epithelium scaling with body size and and there's some information about how many intact receptor genes. And there is an upward trend, but I would caution that you should neither celebrate nor be disappointed by this, because there are also such things as niches. You know, fruit fly presumably likes fruit, and there may be a shaping of the receptor repertoire for the fruit, etc. So this is comparing across many different niches. More care is really necessary, but at least I would say at some vaguely qualitative level, maybe it's kind of okay. But a little bit more seriously, really what we're going after is this these changes in the olfactory receptor repertoire that are known to happen experimentally in the lab. So imagine that you've got two environments. Here's one covariant with this covariant. Let's suppose this is the olfactory receptor, olfactory environment. These are the correlations in the olfactory environment. And a change between this environment and this environment. Here, these two odors have large variance. Here, these two odors have large variance. And you can work either high signal-to-noise ratio or low signal-to-noise ratio. And you find that if you're at low signal-to-noise ratio, you predict fairly dramatic changes in the abundances of different olfactory receptors and the epithelium. So that's what you should predict. What's more, you can do a different thing where you make the same perturbation, right? You take acetophenone, which is an odorant, and you increase its variance or decrease it. But you do it against two different background environments. Right? In the background, it's this or it's that. Right? And then you change one odorant. And what happens is that in a given environment, you get stable, reproducible changes, but the actual result of what goes up, what goes down, etc., depends on what the background environment was. That's because of this global correlation that's there, the correlation matrix. The reason this is important is one of the reasons why I think experimentalists have been scratching their heads about the experimental findings is they find that one lab will be able to reproduce the same stable change, but if you do the experiment again at a different time, you get somewhat different results as to who goes up and who goes down, which then says, well, it's reproducible, but it isn't. And this could be why, because you need to control the background environment to answer the question of whether it should have gone up or gone down according to this theory. So here's the first test of the theory. So there was a paper a couple of years ago by Balasubramanian et al. Where they had mice which they exposed to two different olfactory environments. The way they did this is they basically put the odorants into water. So every time it went to the water, it would get this odorant. A few odorants were increased in variance and so on. And this is the pattern of change they found. This is RNA-seq data. They found that abundant receptors changed reproducibly and not much. Intermediate abundance receptors changed reproducibly and quite a bit. And the ones that were not very abundant, the receptors that were not very abundant in the wild-type mouse, changed a lot. But the change they measured also changed a lot between different iterations. And that's here. This is the theory down to 60 mouse receptors. That's all we have, and a model of this environment, and you see the same pattern of change. So I would say you could test this if you, of course, by doing some environment engineering and having more detailed data about these receptors. There was this Olfaction Ideas Lab a couple of years ago in which two of the groups promised to completely measure the entire mouse repertoire and its responses to many odorants. Another group promised to measure entire olfactory environments. And if you have that data, we will do a check. Great. So how many minutes do I have left?
7.
7 minutes. Okay. So I'm going to try to go through. Let me try to go through this next. So this is a little bit of a way station of the way to what I really want to get to, which is the decoding. And I want to tell you about, I've been sort of approximating everything as linear sensing. And I want to convince you actually we can do a bit better than that. So let me just go ahead and say that. So real olfactory sensing isn't the simple linear picture. It's more like this, things bind to binding pockets. Well, there's a very long history of people trying to biophysically model how molecules bind to different binding pockets and stuff like that. And the most elementary nonlinearity you're going to find is that if the green molecule binds, the red one can't. And this is called competitive binding, and it's going to produce nonlinear effects in the overall responses to mixtures, right? So that's the minimal thing you could imagine. So you could ask, will this minimal model already get me some distance of the way to understanding, the responses to olfactory mixtures? And here I'd like to just show you some data. Yes, that's true. Here are many receptors. There's like 15 receptors, many binary and ternary mixtures, as you work out this kind of competitive binding model using dose response curves like this, and you get a very good fit. This is the best model on the market. It comes within 15% for basically all the mixtures we tried. So that's good. I'll take questions at the end because I'm really running short on time. Okay? So What's more, if you go to more real odors, like 50 components, and there's no experiment that tries different combinations of 50 components. But our collaborator measured different combinations of 12 odorants, at least it's getting some distance towards 50. And now still we find an excellent fit to the data. If you try the linear model, you'd be like 100-fold off compared to this. Right? So this is a very significant improvement, dramatic improvement on my default model inside my head, which is linear sensing. So you can do definitely better than that. And I'd like to say that we've numerically tested some of the statements I made in the previous part of the talk with this kind of more sophisticated model of sensing, and things go through in a similar manner. So the remaining 5 minutes, 4 minutes, 6 minutes. I'm rich. In the remaining 6 minutes, I'm going to tell you a story that we've been trying to tell lately on decoding this kind of adaptive combinatorial code. Right? I said in the beginning that if you really believe, if you're a compressive sensing person, you compress the odors into the small space. It's an algorithm, and you can implement the algorithm and decode these odors. But the relevant algorithm is pretty complicated. Well, it's easy to implement in Mathematica, but it'll be a little bit of a mess to implement it in the brain. So here's an idea. So I want to know how these combinatorial responses can be decoded. And it turns out it's an exceedingly simple approach to decode them. That again depends on the sparsity of odors in the real world. Here's the way it goes. We call it estimation from elimination. And I can describe it with the schematic. So here, permit me to talk about binary receptors. This is like a high noise situation, right? Where if you have an odorant, the receptor response, right? Response of 1 or 0, right? High threshold for measuring things. So here's a random sort of looking sparse sensing matrix of the kind we think we have. Here's a sparse odor vector. If this sensing matrix senses this odor, and here I'm still talking about linear sensing, we'll correct this in a moment, then these two receptors will respond. Well, suppose I tell you these two receptors respond. Please tell me what the odor was. What's the problem? The problem is the stupid thing is underdetermined. You have fewer responses than you have odorants. Now comes an interesting point. Normally, the way we're used to thinking is the receptor that responds tells you something about the odor. But actually, it's the receptor that doesn't respond that tells you more. Here's why. So suppose this receptor doesn't respond, like the first one. It tells you that this odor, or this molecule, and this molecule must be absent. This non-response tells you this molecule must be non-absent. So you can very, very quickly eliminate just about everything with a few non-responses. And then you'll figure out what's left is what the odor is composed of. So you can ask, would that really work? So you can do a quick statistical estimate, and you'll find the probability of making errors depends upon this quantity, e to the minus the sparsity of responses times the number of receptors times the exponential sparsity of responses times the number of odorants. Let me say a couple more words about that. So what this tells you is that for good performance, the sparsity, maybe the fraction of things that respond, times the number of receptors should be large, so that many receptors respond to an odor. So that's to ensure coverage. Right? On the other hand, the sparsity times the complexity of the odors, right? How many components are in an odor, should be small so that there are sufficiently many inactive receptors so that you can figure out what's not there. And you can plug in the kinds of numbers for Drosophila and human, etc. And so, for example, if you take as order of magnitude 10,000 odorants, this is a little bit backward here, 10,000 odorants with maybe 10 components per odor, order 10, 500 receptors, something like that, sparsity of 5%, you'll find that the probability of false positives is 10 to the minus 7. So there's a lot of room here in these ballpark numbers for relaxing things so that things will work out better. Right? So things will work out well. So let's push this. So in view of the time, I'm just going to put up this plot that tells you that you need to do this numerically, this, that, the other, and you'll find that there's a large range of complexity of odors and sparsity times number of receptors where things go well. The reason I want to move on is really because now we have this competitive binding theory that's supposed to give us a model for the nonlinear responses. Okay, so I'd like to tell you that the same decoding algorithm, with a slight tweak, will also work beautifully for that. So once again, I have this sort of encoding process where I have the odors getting encoded, and then by some sensing matrix. Really, I don't mean sensing matrix anymore. I don't know how to draw the full nonlinear response of these things, but there's some response that comes by the receptor sensing the odors. So once again, we can do the following thing. Let's suppose that some receptors do not respond to some odors. Then you use the same elimination mechanism to rapidly eliminate lots of odorants that are absent. Right? They're not there. And then you're left with the problem where you have the actual sensing mechanism, an unknown set of concentrations, and the responses that you actually see. And this becomes a well-defined inversion problem. You have to solve a nonlinear problem of solving, how you go from this response non-linearly, these inputs, but it's a well-defined problem that can be solved with some equation you can write down, and you could turn this into a neural network if you wanted. So I'm going to finish with that network. So once again, here's a cool thing. If you again trace out the parameter space of odor complexities, number of receptors, etc., turns out that the operating point at which you get the best performance for the 50, 60 component odors is when S times K is about the S times the number of receptors. The sparsity times the number of receptors is about 10. And if you work out these numbers, for humans, it's like 12. For Drosophila, it's like 9. Right? It's about exactly where it ought to be for being around here. And so I'd like to suggest that perhaps, again, this explains the operating point. Why you have a few hundred receptors with a certain degree of sparsity in the responses. Right? Because if you were doing that, you'd be able to decode very, very well. Right? So perhaps that's the case. I will end by saying that there is a neural network implementation of this algorithm, which really resembles, at least broadly, the kind of architecture you see in the olfactory pathway. What you would need is something responding to the concentrations of odors. You would need some summation to beat down the noise. You need some lateral inhibition to kill off those responses that really should be zero. Right? You don't want any noise kicking a zero response into finite response, right? Because zero matters for us, right? Non-response, more important. So that would do that. And then you want to go to the cortex, where again, if you write down a decoding algorithm, if you rewrite the decoding algorithm into a neural network, it would look like you would need some feedback inhibitions. I don't have time to go through that. So I'm out of time. So I'm just like, this is not a claim that that's what the brain is doing, right? I don't know. But it's a claim that the architecture is what you would need to do this kind of decoding, especially because if you look at given cortical neuron. So the interesting point is that if you look at projections to the cortex, you find that every cortical neuron collects the responses of many types of receptors, but each of these synapses are weak, so that if any one of them doesn't respond, the cortical neuron doesn't respond. That's exactly the kind of thing you want to do here. If you think about a cortical neuron as kind of trying to decode an odorant, it should only respond if all the receptors that bind to that odorant respond. If any one of them doesn't respond, you should kick out that odorant as a candidate. So the growth structure does look like that. That'll end there. So the insight here is that the receptor that does not respond carries a lot more information than one that does. There's a series of predictions you can make for what could happen after optogenetic representations. So anyway, the title of my talk is Becoming What You Smell, and that's because I think all of these things, these stories about adaptation of circuitry in the brain to the world, is kind of a statement that in this case the nose becomes a representation of the world. You mold the geometry of information of the circuit inside your nose to the geometry of the information of the world. Okay, I'll stop there.
Go ahead. We'll have one question.
Yeah.
No, go ahead.
Oh, okay. Totally fascinating. Would your approach extend to understand the proprioceptive and haptic system that has an additional active spatial configuration, including that some of the sensors can be tuned, and the number of different aspects that contributed, I suppose you know.
Yeah.
So could you extend, or is it extend your approaches to understand that system?
Yeah, so I have not thought carefully about that, so I can't give you a completely meaningful answer. But I think the more I've been thinking about this thing, I've got the feeling that in the end, the kinds of things that we need to represent are contexts for behavior. And we always have the situation that the possible contexts that you have, whether it be haptic or whether it be auditive or whatever, are This is a huge space. There's many, many different types of contexts, and it's a very high-dimensional space. But the actual context you get is, in some sense, a sparse vector of that, right? Because most things are not happening at any given time. So there is, well, it seems intriguing to consider the possibility that this kind of view, that what you should do at some point is you sort of randomly compress the information and then decode it in this kind of manner, might help. I can't present any detailed evidence in any other part of the brain, but then I haven't thought about it carefully. It'd be interesting to think. The hippocampus would be one interesting place to think about this.
Yeah. All right.
Oh, yeah.
Sorry.
They have this, the USB-C.
Oh, yeah, the mini? Okay.